don't mind, we have a lot to cover today. I know some of you have a neuroanatomy midterm and everything else, so I'd like to get going if that's okay. Okay, folks, let's go. All right. Okay, I actually really want to get going. We have a lot to cover. I'm going to talk to you a little bit about your individual term tests, the mechanism that we're going to try to do, because some of you might want to ask for a regrade. I hadn't thought that all the way through, to be quite honest. I've returned as much as I can to your groups today. So you have, many of you have your group assignment. You also have your group term test and everything else. And some of you have got your marks up already. For those of you who just got it back from Victoria, I'll be posting your marks. I have the spreadsheet, but she just returned the assignments literally a few minutes ago. So I'm going to take you through this. I'm going to talk to you just before the break about how we're going to do the individual term tests, picking them up. And if you're going to ask for a regrade, I'm going to ask you to make sure that you see the group assignment first, because you have a good indicator of what I'm looking for in terms of the group test. And if you have anything that you want to get regraded, I'll be happy to take a look at it. No guarantees that it'll go up, down, or change at all, but we'll see how things go. I'm going to talk to you a little bit about different aspects of sleep. And this is actually not a trivial thing. As neurobiologists and as geneticists, sleep is something that we don't really understand. In fact, at U of T, just in psychology and in cell and systems bio and physiology, we have 4 separate sleep courses. And sleep is something that we really don't understand. We go through a lot of different things that we intimate about sleep, but we actually don't know as much about sleep as we'd like to actually reveal. And so today I'm going to go back and talk to you a little bit about one prion disorder that is related to sleep. It is an autosomal dominant disorder. It is a fatal disorder, and we'll go through that fatal familial insomnia again a little bit later on. But before I get into that, I'm going to talk to you about prions a little bit. I'm going to talk to you about sleep. I'm going to talk to you about other disorders of sleep where you're sleeping too much, not you're not sleeping enough. And then we'll get back into prions, and we'll make use of what we've learned previously about prions in today's lecture. I don't know if any of you actually saw this issue of The Scientist, and it's not important for you in terms of the midterm, but your final exam, but in terms of just understanding sleep. People have been trying to study sleep for a very, very long period of time. This one paper that came out of Yan Dang's group actually was really interesting in the sense that in the old days, one of the things that psychologists as well as neurobiologists used to do was to just simply ablate, take out different areas of the brain and see what the function is. Now, if you take a look, this is one little area of the brain. And in this one little area of the brain, you have glutamatergic neurons in the forebrain, you have cholinergic neurons outlined in blue, and then you have these somatostatin-containing neurons, and you have these parvalbumin-containing neurons. That's all in the same area. So imagine you're just like taking out this little section. So what is really good about genetics and genomics right now, just as we've learned, you can actually go in and use different drivers and different promoters to actually stimulate specific groups of neurons. So you stimulate the glutamate groups of neurons, and you see which neurons get activated using electrophysiology. And what you see is when you stimulate glutamatergic neurons, and only glutamatergic neurons using optogenetics, you then see the activation of other cholinergic neurons. And these cholinergic neurons in turn will turn on the somatostatin-containing neurons, etc. We still don't understand the entire circuitry of sleep because it's really, really complicated, because it's not where you can just take out this area of the brain like we used to think and understand the mechanisms that control sleep. But it is something that I want you to have an idea about, why we're worried about sleep, why sleep is essential for us, and why we haven't made that much progress. We've been looking at sleep for over 150 years as geneticists and as neurobiologists. We have 4 different courses on that here at U of T, but we still don't understand everything related to sleep. And part of it is because it's really, really complicated. So I talked to you about prion diseases, and prion diseases are really interesting. We can have it as a transmissible form. We can have it where we're intaking something that has a scrapey version of a prion protein, and that causes our own prions to become misfolded. We go from alpha helices into beta pleated sheets, and these beta pleated sheets become really insoluble, which then cause neurons to start to degenerate. And these are, generally speaking, progressive. You never get better once you have a prion disorder, and they're generally fatal as well. We also know that there are some inherited versions of prion disorders. So we'll be talking about fatal familial insomnia, and there is the Gerstmann-Straussler-Schenker syndrome as well. And these are the most common forms that we see in humans. Some of these are inherited. Some of these are also transmissible forms. And then in animals, we know that there are zoonotic factors as well, and prion diseases that affect specific animals like goats, sheep, as well as in cows, mad cow disease or BSE. All of these things suggest that these things are around, that in some instances, there's some suggestion that we've adapted to the environment where we find these different prions. But these genetic prion diseases are the ones that are really interesting for us, because they also tell us a little bit about this sleep circuitry. So again, when we start looking at what's going on in the brain, usually we see this neuronal loss. When we start accumulating more of those beta pleated sheets, and we start losing the cellular normal form of prions that we all have, we start losing neurons. We have reactive gliosis. There are spongiform changes. There are literally holes that exist within the in the brain as well, or in the neuropil. And again, we sometimes see what looks like amyloid deposits. And so when we start seeing these prions and amyloid like fibers, one of the things that starts to appear, and it's universal, it's something that's common in CJD or Creutzfeldt-Jakob disease, or it's common in FFI, as I'll show you in a moment. For whatever reasons, you start to lose motor coordination. There are other motor abnormalities associated with prion disorders as well. There are often cognitive consequences, even though it's not up in the gray matter to begin with. It has some cognitive consequences that I'm hoping to show you in a few moments. So again, we went through this really quickly, and I'm not going to spend a lot of time on this. One of the things that we think this is a precursor for is that it gives us an idea that in the nervous system, when we have aberrantly folded or we have issues with transporter proteins, that there might be something that causes neurons to start to die. And we'll come back and revisit that theme in a little bit. We also know that we can acquire this by infection. It doesn't have to be only by eating. You could have an infected surgical instrument, for example. You could be injected with someone's serum or someone's hormone that's been infected. And so when we talk about human material being passed from one person to another, however it's being passed, we can also have these prion disorders that can be acquired through infection. And again, it's usually that scrapey form that causes the infection. And at the end of the last lecture, I tried to convince you that one of the things that we think happens is that maybe, just like prions, that some of the other proteins that are associated with neurodegeneration are also passed along in a very similar manner. Okay, so one of the things that I did not show you in the last lecture, and it's important for you to maybe go back and review at the end of this lecture, and it's something that you probably will see on your final exam, is that when we talk about prions and the inherited forms of prions, oftentimes we see mutations occurring in prions. And these can be spontaneously occurring, which are then transmissible or hereditary and passed along to the offspring. And oftentimes we see these insertion deletion types of mutations. And oftentimes when we look at chromosome 20, where we find the prion protein, we see that these mutations tend to be clustered in the C-terminus region. And I'll show you this diagram again in a moment. We know that sometimes these insertions also occur, especially in things like GSS, where we have an octapeptide repeat in the N-terminal region, and that could be also an additional insertion as well. So for whatever reasons that we're not entirely sure of, even now, that when we have these different mutations that are associated with things like fatal familial insomnia or CJD or other prion models, that the inherited forms tend to spontaneously develop, but they seem to have a very, very, I would say, well-defined sort of timeline. So it doesn't just occur at any old time. It occurs often when you're in your 40s or in your 50s for whatever reasons. And to be honest, we still don't understand why, for example, if you have a point mutation, it takes that long. You're in your 40s and 50s before you start to have motor abnormalities, cognitive deficits, prion diseases that look like familial insomnia, etc. So there's still a lot, in other words, that we don't understand about this. So I wanted to draw your attention. In the last lecture, we sort of looked at both of these sort of as a model, like a misfolded protein, something that gets messed up in how it gets transported out of the cell, or it might be something that accumulates inside the cell. Whatever it is, there seem to be some similarities between the beta amyloid at the top and the prion protein down here at the bottom. And these are sort of the octapeptides that I mentioned to you earlier. Sometimes we have additional copies or insertions that are put here in the N-terminal region, but oftentimes we see the greatest number of point mutations, either insertions or deletions down here near the C-terminal end. One that's really important to us, in fact, there are two that are really important to us, is at position 129, and then there's another one here at position 178. For whatever reasons that we're not 100% sure of, these are ones that seem to be inherited the most. And when we have a mutation at position 129 or at 178, this often, when they're in combination with each other, are often very, very deleterious. And again, there's this core fragment that we see, just like we do with A beta, that seems to be really important in terms of how this protein misfolds. And you can see that the point mutation is actually outside of this core region within the prion protein. So the prion protein core region is outlined for you here in red, and the core region for beta amyloid is outlined for you in red here, and that's the beta amyloid fragment. So there's this core thing that starts to misfold and starts to produce these beta pleated sheets. It's outside of the point mutation. We don't really understand all of the reasons for it. It might be interacting with other proteins, etc. But I wanted to just have you keep that in mind for the time being. So again, when we look at different types of mutations that are associated with different phenotypes, it's really fascinating, right? So if all these different things cause basically the same protein to misfold, why does the disease look so different? So fatal familial insomnia looks different than GSS, which looks different than Jakob-Creutzfeldt or Creutzfeldt-Jakob disease. And all of these things may be targeted to different regions of the brain, and we don't understand that. So for example, we have abnormal prion protein accumulation in the thalamus for FFI, and with CJD, we have abnormal prion proteins. The same sort of prion protein is abnormally folding and forming beta pleated sheets in different parts of the cortex and parts of the cerebellum. It's not that they don't go everywhere over time, but this is where these tend to start. So we also know that we might develop systemic amyloidosis as well, and that this is that codon that seems to be really important. So whether or not we have a methionine or a valine at this codon 129, and in addition to that, something also occurring in codon 178 makes a real difference in terms of how toxic this prion is, as well as how it presents itself. So if you have one version, it's going to present, even if you have a mutation at codon 129 and you have a different mutation at codon 178, it will present as a form of Creutzfeldt-Jakob disease. And if you have another variation, it will present as fatal familial insomnia. Okay, so again, the other important codon is this 178. So depending on what happens, whether you have a mutation that goes along with 129 and 178, you will either have fatal familial insomnia or you'll have a variant of Creutzfeldt-Jakob disorder. And again, there's very, very big differences in terms of how this phenotypically looks. So when we look at a mutation at 129, where we have a methionine at 129 and then we have another mutation at 178, this is fatal familial insomnia, which I'll be spending most of the time talking to you about. and accompanied by things like autonomic dysfunction. And the Creutzfeldt-Jakob variant is where you have a valene at that position, even though you also have that same sort of variation at 178. We don't understand why that occurs. We don't know how it targets one to the thalamus and the other to different parts of your cortex. It's still largely unknown. And maybe one day we'll have a better idea of the pathogenesis, but right now it's a black box. We just can identify what we see more commonly associated with FFI or with CJD. So we don't understand this variability. We can only describe the phenotype and then look at the accompanying genotype that goes along with it. So again, when we take a look, in terms of FFI, we see most of the abnormal prions being built up within the thalamus, and in CJD, we see very similar types of histopathology where we see all this vacuolization. So these vacuoles, these large open spaces that we see are primarily found throughout the cortex. So we still have two mutations in prions, but they're targeting different regions of the brain, causing dramatic differences in the phenotypes that are being presented to us. Any questions about that before I go on? No? Yeah, Eddie? Sorry? So actually, we see basically in both the histopathology leads to vacuolization. Like we see big holes. It doesn't matter which one you're looking at. All prion disorders have these large vacuoles that seem to exist in the neuropil. So that would be representative of both. So either or both, yes. Okay. So when we take a look at some of these different mouse models, and I'm just presenting this to you because we need to understand this disorder. People have tried to create different transgenic models. I'm going to show you another one a little bit later on. So the most commonly used background mouse model is this PG14. So this has that octapeptide repeat. There's an extra octapeptide in here. There's an insertion, in other words. This is often associated with that GSS, which is an inherited form. And then in addition to that, it has this sort of other additional mutation associated with it, which is the most common type associated with CJD. So again, those two different amino acids. If you're wondering about the specific numbers and things, don't worry about it. In terms of memorizing the numbers, there are slight differences between mouse and human, and we'll see that again a little bit later on. So one of the interesting things about these particular prions, when you have this mouse and you take a look at the prions, if you were to tag them and see where they're located, these prions tend to be accumulating in the endoplasmic reticulum. Now just keep that in mind for the time being. So the endoplasmic reticulum is really important in the maturation of different proteins. So it goes from ER, Golgi, and then gets shuttled into whatever compartment it needs to. And these prions that have these different mutations, whether it's an insertion and then substitutions or point mutations, these particular prions in this P14 mouse, PG14 mouse model tends to accumulate within the endoplasmic reticulum. And when these proteins and these prions accumulate, they also cause other proteins that are normally trafficked through the ER Golgi to also aberrantly accumulate as well. One of these happens to be this voltage-gated calcium channel subunit, the alpha-2 delta-1 subunit, and as a result, it doesn't get to the presynaptic terminals like it normally should, and it gets stuck in the ER as well. So the prions get trapped there, but in addition to the prions getting trapped in the ER, so do these voltage-gated calcium channels. So what ends up happening is that wherever this CJD mutation occurs, that we don't have really effective glutamatergic neurotransmission, particularly in cerebellar granule cells. It also affects parts of the motor cortex. And as a result, we see this motor impairment in these mice. So this is actually an important sort of observation that different scientists started to make. So we still don't really know what the pathogenesis of prions are. So why do we have neurons dying if we start having these beta-pleated sheets? Lots of proteins are beta-pleated sheets. Why are these beta-pleated sheets causing these neurons to die. And so one of the interesting hypotheses that has come out recently, in addition to misfolded proteins as one model, is that overall, if you take a look at neurodegenerative diseases, there's also a cohort of neurodegenerative diseases where there seems to be intracellular accumulation of proteins because they're improperly trafficked, or they're not properly let out of the ER into the Golgi. And so this ER retention might actually be the beginnings of why we get the phenotypes that we do. And this is sort of the key question. Is this sort of a universal mechanism that explains why prions accumulate inside the ER, how it kills neurons, and then why it causes neurodegeneration? We haven't really seen a lot of that in FFI, so that characterization has really been in CJD. Is this also occurring within FFI? So again, keep these hypotheses in mind for the time being. So this is what we're going to be talking about, this autosomal dominant disorder. So you see that every generation is affected by fatal familial insomnia, where we have mutations in the prions. And again, this is also spongiform. We get vacuolization. The brain overall looks like it's got large vacuoles within it. It tends to be lethal depending on the variant that you have. You could die as soon as 6 months, or you could die a year and a half later. Usually it's not a very pleasant sort of death that you get either, and hopefully I'll be able to show you that in a moment. And usually before you die, so what happens is this disorder is really, really bad. In fact, if you go online, you can look this up later. I'm not going to show you this particular video, but the video that I was introduced to when I started in grad school was the video of Michael Cork. And Michael Cork is this musician and a music teacher in the United States, and he develops FFI. And if you see what happens to him and his slow degeneration over the course of 6 months, so he passes away after 9 months, and you might think, okay, not being able to sleep is not such a big deal, right? So what would you do, for example, if you couldn't go to sleep, which is probably not your problem? You have the opposite problem of being able to stay awake. But what would you do if you couldn't go to sleep? Yep. Take Gravol? Okay. Interesting. Gravol. Never heard that one. Okay. Yep. Finish your assignments a lot earlier. Okay, sure. Listen to one of my recordings, also very useful. Yes? Okay, melatonin. Okay, all of that. So how about I tell you this, that when you first starting having the first episode of insomnia, and you can't go to sleep, and you can't go to sleep after a few days, actually taking a sleeping pill, taking some sort of benzodiazepine, which is what a doctor would normally prescribe you to get you to go to sleep, actually makes this worse, and actually makes the whole sleep architecture, how you go from REM, NREM to REM, etc. It makes the architecture a lot worse, and it actually speeds up the process. Oftentimes, again, because you have all these other things going on, you have like paranoia because you can't sleep, and there are sleep disturbances that are associated with things like multiple sclerosis, that oftentimes it's misdiagnosed as multiple sclerosis. And eventually, over time, you have psychotic episodes, you might have issues. And again, if you see Michael Cork's video, which I'm not going to show you, he has dramatic weight loss associated with it. They have very characteristic motor movements. They have issues with their autonomic functions, as you'd expect. And again, this is a relatively late onset. And this is what makes this disease really, really bad, right? Because if you're in your 40s, you're likely already going to have children. And if you already have children and it's an autosomal dominant disorder, it's likely that half of your children have the risk of developing this disorder that you never knew that you carried. And so this is sort of the issue, and this is sort of what we're going to be talking about in terms of FFI. It is not a pleasant way to go, and I'll hopefully show you this in a few moments. So let me take you through a couple of things before that. So let me talk to you about sleep in general, and how we need sleep, and how we think that it's all circadian rhythm generated, how it's blue light dependent, stuff that you had in your third year or PSL 300 courses. You don't have to worry too much about more than what I'm going to be talking to you about here today. Let me ask you, when you have not slept for a few days, what happens to you when you go back to sleep for the very first time? So you've stayed up, let's say you're really plugging your way through a bunch of midterms and everything else. You stayed up for maybe 48 hours. Some of you might have stayed up for 72 hours. If you saw my post earlier on, the longest recorded episode of not sleeping, not because of a disease, is 11 days, 264 hours. That's the maximum recorded time. It was done at Stanford University many, many years ago. Other people have claimed to have actually done this. The Guinness World Book of Records actually doesn't keep track of it. They don't want people to not sleep. And so the longest recorded, officially recorded time that was done in a scientific way is 264 hours. So when you've stayed up that long, you have what's known as sleep rebound. You sleep a lot longer than you normally would. If you're used to sleeping 8 hours, you might sleep like 18 hours, you might sleep 24 hours, etc. We know that in mammals, the suprachiasmatic nucleus is really important in setting this whole train of sleep. And we also know now, in retrospect, after the 1980s, that neurons and groups of neurons within the hypothalamus that are part of this whole sleep architecture, neuronal architecture, are really important. So we don't really know, as I mentioned from the outset, what controls sleep. We don't know whether it's metabolic. In fact, we don't think it's metabolic. You can control the metabolic processes, but we don't think that metabolic factors are what cause you to go to sleep, etc. There are probably lots of things. So let me again just take you through this one particular slide. It's more psychology than it is neuroscience, but I always am really fascinated by this. So if you take a newborn infant and you look at their period of sleep, they'll go in and out of sleep all the time. Like they're used to sleeping and they'll go in and out of sleep, and this is sort of the sleep pattern that they normally will have. After about a year, they're still going in and out of sleep, so those sort of naps that they take in the middle of the day if they need a nap, but they are sleeping more at night. So they're being entrained, in other words, and this becomes a theory that maybe there's some entrainment of neurons that lead to this sort of sleep cycle that we have. And by 4 years old, we're sleeping in the afternoon a little bit less, and by the time you're 10 to 12 years old, you're sleeping more or less like an adult, where you're going to sleep at night and you're waking up during the day. And we don't, again, we don't really understand the full mechanisms of this. There are lots of theories to try to explain this as well. If you, for example, have one particular disorder that I'm going to be talking to you about, so we're going to talk in a few moments about the lack of sleep. That's FFI. There's another genetic disorder, which is where you are falling asleep at inappropriate times. Do you guys know what that disorder is called? Narcolepsy. So narcolepsy actually has the exact opposite patterns. You're usually used to sleeping at night, but narcoleptics usually have excessive daytime sleepiness. They might have prolonged drowsiness, and if they have the really extreme form of this, those narcoleptics will fall asleep at any time, right? And it seems really funny, but imagine that you're standing right there at the subway station, the train's coming in, and at that inappropriate time, you fall asleep and you fall onto the tracks. Usually doesn't happen, but in theory, it can, because going along with the narcolepsy, where you have this really extreme sleepiness, drowsiness, you also have associated with it another disorder called cataplexy, and that goes along with sleep, right? So during sleep, your muscles, your skeletal muscles tend to relax, and when you start going to sleep with narcolepsy, you also have this cataplexy, where you suddenly will faint, or you'll lose muscle tone, and that cataplexy is also there. Oftentimes, if you want to trigger someone, you shouldn't, but if you were to do this, it's often associated with an emotional reaction. They feel really scared, or they feel surprised by something, and a narcoleptic will suddenly lose muscle tone and start to go into sleep right away. They will have this sleep paralysis, but they will also have a very abnormal REM and EEG that goes along with this. Oftentimes they have hypnagogic hallucinations, and they have vivid dreaming episodes. Their dreams are not like your typical dreams that you and I would have. In some instances, there seems to be a loss of memory, and for whatever reasons that we're not 100% sure of either, if they carry the genetic background for narcolepsy, these individuals will start to begin between the ages of 10 to 25. And again, this is sort of where the entrainment occurs anyways, and it's thought that that is somehow related to when this particular narcolepsy occurs. So we know that there are lots of animal models that actually have this spontaneous form of narcolepsy, just as I mentioned to you in humans, particularly Dobermans as well as Labradors. Some breeds of these dogs have spontaneous narcolepsy. But this has actually been really beneficial to us. Remember at the very outset of the course, we talked about mouse models, and we also talked about, in Duchenne's muscular dystrophy, about dog models for this. And usually we don't work with larger animals. We don't work with dogs because they're more expensive. They tend to have a number of other issues associated with them. But here is actually where, in neuroscience, this has become really incredibly valuable to us as neurobiologists as well as geneticists. So I don't know how much of a neuro background you have. I'm hoping that some of this is somewhat familiar to you. Do you guys remember what the word orexin means? Orexigenic? Orexin, or also called hypocretin. Do you remember what orexin is? If you're anorexic, that orexin. What is orexin? So it's a hormone associated with hunger and eating and everything else. And one of the things that was found back in 1998, that in certain groups of neurons within the hypothalamus that we still talk to you about today in physiology, that there were a group of neurons that were identified by two different groups. One group called it orexin, another group called it hypocretin. It turned out to be the same protein or the same peptide. And so it's orexin / hypocretin, because two different groups came up at the same time with the same molecule, and it turned out to be a neurotransmitter. And it is. It's really important in things like eating behavior, and this is why we call it orexin. So when we stimulate these neurons that contain orexin, you stimulate feeding behavior. So people wanted to know, is this really that important in terms of your control of feeding? And so as geneticists, one of the things that people did, and this came out in Cell in 1999, it made huge news because this molecule that was associated with eating in the hypothalamus was actually, when it was knocked out, actually caused those animals, and these are knockout mice, to spontaneously fall asleep. They exhibited narcolepsy, in other words. So a neurotransmitter that was supposedly involved in something like eating and only for eating actually turned out to have multiple functions. And in this case, it was important in keeping you awake, because if you knocked it out, it caused these mice to have narcolepsy. And these are where these papers came out back to back to back in Cell and in Neuron and in other places where in orexin or hypocretin knockout mice, they exhibited narcolepsy. When you go back in and take a look in those canine models, spontaneously occurring narcolepsy, when you look at why they have narcolepsy, it turns out that there's a problem with the orexin receptor. So again, the same system, suggesting that orexin was the molecule that controlled sleep. And when you looked at human narcolepsy, which came out a short time after, about 6 months after that, there was a reduced number of these hypocretin neurons in individuals that had narcolepsy. So we thought at the time, back in the late 1990s, that we understood this circuitry that was involved in sleep-wake cycles, and it was all related to orexin as well as hypocretin. That was sort of the big dream. It turns out that it's much more complicated than that. And again, these narcoleptic dogs had these mutations in the hypocretin receptor, which is why they didn't respond and why they fell asleep. And the hypocretin receptor turned out to be a G protein coupled receptor that they found through positional cloning against other GPCRs. And again, when they look at people with narcolepsy and in human studies, they also saw that there were fewer of these hypocretin-containing neurons within the hypothalamus. So it suggested, and it was sort of a done deal, this is how we fall asleep and this is why it's important, that hypocretin and orexin were involved. So again, it's a very small number of neurons that actually produce orexin. Not many of them actually do. There are two different variants, hypocretin 1 or orexin A, and hypocretin 2 or orexin B. And again, sorry about the nomenclature. It's still equally used by different groups. Some people like orexin more, some people like hypocretin more, but it is believed to act transmitter, and I'll show you where it spreads out from the hypothalamus in a moment. We had talked about looking at relative risk ratios when we started looking at things related to schizophrenia. If you look within medical literature and you want to see an association between a disease state and a particular a particular SNP or a particular association with a gene, these are actually the highest relative risk ratios in medicine. So if you look at someone who has narcolepsy, they're likely going to have one of these different mutations associated with the human leukocyte antigen. So if there's something wrong with their immune system, which makes sense because it's thought that in those individuals that have narcolepsy between the ages of 10 to 25, that somehow their immune system gets activated, they turn on autoimmune antibody-mediated destruction of neurons, and that's why when we look at their brains, that overall they have less of these hypocretin-containing or orexin-containing neurons. And it's likely because they are overexpressing some of these human leukocyte antigens. So again, we won't worry about all of this. This is what a normal hypothalamus, where we have all of these little dots represent a positive stain that you see here. And this is that little box that you see here blown up to show you the individual neurons, and these are the neuronal cell bodies all staining for hypocretin or orexin. And this is in the narcoleptic individual. You can see that there are many fewer of these orexin positive groups of cells. So we don't have as many, and this is probably the reason why they fall asleep at inappropriate times. This is that small group in the lateral wall of the hypothalamus toward the ventral or posterior region of the hypothalamus, where we get the greatest concentration of these orexin-containing neurons, and they spread out everywhere. They send their connections to a number of different places, which is probably why we haven't understood why they control sleep as well as we originally thought we would. I'm just going to mention this one experiment. It's something that I want you to be familiar with if you haven't heard of this before. How many of you take biochemistry? Have you heard of saporin? No? If I want to kill a cell, there are lots of ways for me to kill a cell. Neurons in particular have a lot of Golgi. They're really protein building machines. They need a lot of this protein machinery. There is this toxin known as saporin, which actually blocks the transport from the ER to Golgi. It's an ER toxin, in other words. When we put, for example, saporin on molecules that track to orexin-containing neurons, if these are orexin-containing neurons, the light blobs that you see up here, and then we attach saporin to specifically attack hypocretin-containing neurons, so we could have a saporin hypocretin receptor, which will target the hypocretin containing neurons, then you can see that they get completely wiped out. So when you wipe out these neurons and then you start looking at the behavior of the mice after wiping out these neurons, the greater the number of neurons that you have, again, the less overall sleep that you have. So if you don't have hypocretin containing neurons, then you are going to be disrupting sort of the sleep that we talked about a little earlier on. So this is one mechanism that I want you to be familiar with. And there's also, yeah, Danny?
Earlier you said that when they found that the proteins stay in the ER.
Yeah.
Is that at all related to this?
It may be. I'm going to come back to this because I want you to know that this toxin, saporin, it actually blocks that ER transport as well. And this is one way to actually kill neurons in a specific way. You can tag these saporin molecules and kill specific groups of neurons. In this case, you're killing neurons that contain hypocretin or orexin, and you can disrupt their sleep by doing so. Okay. So the big question is whether or not prions also play a role in sleep. And I'm going to see if I can get this thing to work. I'm hoping it does. Okay, folks, if you don't mind, just so I can get you out of here for your bell ringer, for those of you who are doing neuroanatomy on time today. And we'll try to also make sure that you know what the expectations are for your group presentations as well. So I'm going to show you a really quick video. We're not going to go through the whole thing. That gives you some ideas about why FFI is really such a scary disorder. You'll get to see some very characteristic types of movements that are associated with FFI. I also teach a course in personalized medicine, and it's always really surprising to me, like if you think that you have a genetic disorder, would you get tested for it or not? And it's always really surprising to hear how different individuals actually go about deciding whether or not they are going to or not. And so we'll just go through this real quick. Sorry about the lousy music.
It's my story. I get to choose whether I laugh or cry about it. It's my choice. I'm gonna laugh about it. It's my day. It's my life.
Carolyn and Cheryl are sisters who have lived for years with a mortal secret. Like a poison fruit, a genetic mutation hangs on the family tree. It killed their grandfather, their uncle, and in 1999 revealed itself in their mom, Barbara.
Your mother saying, Mom, what's wrong? Why can't you speak?
She couldn't speak because she could not sleep at all. Inside Barbara's brain, a genetic tripwire had been crossed. In a matter of months, she went from being a vigorous 52-year-old woman to a coma, emerging only a few days at the very end.
They took her intuition out, and she couldn't really talk because she was so dry-throated and everything. And she was trying to write, and she wrote FFI with a question mark behind it. And I said, that's what they think it is. And she just kind of went, she never wanted to think it. She never wanted to think it to happen.
FFI, fatal familial insomnia, is an
So you'll see the motor disorders associated with it.
Victims' sleep, their mind, and their life. It affects about 40 families worldwide. Barbara didn't know it when she died, but odds are 50-50 she passed it on to her daughters. Before this story is over, we will know which one of them has decided to learn her fate and which has chosen to remain in the dark. But to understand this disease, we must start in Italy, 250 years ago.
Yes, it's actually a really cool. It's a really cool story.
Unknowingly carried the original genetic mutation for FFI. But the experts simply refer to him as patient zero, the first known case of the disease. By the time he died in 1765, he had passed the gene and the curse to his children, and the chain had begun. In its first stages, family members experienced a kind of insomnia we'd all recognize, but those sleepless nights never ended.
I'd say within a month, it's pretty clear that you've got a disease like no other disease.
D.T. Max has spent years researching the phenomenon of FFI. His book, The Family That Couldn't Sleep, traces the history of the Italian clan who first carried this cruel disease.
1, you're not sleeping. 2, you're having difficulty walking. 3, your ability to focus has gone downhill rapidly until about ordinarily the 9th month the disease ends with death.
Many of the afflicted Italian family members were brought here to San Servolo, an island asylum, just a short boat ride from Venice. Now a museum, San Servolo remains a haunting place.
What would it have been like to walk inside these walls?
There would be howls, there would be people doing uncontrollable behavior, there would be attendants chasing after them.
I saw one room downstairs with leather restraints. Some of the treatment was simply just keeping these people from
That's right.
hurting each other.
Generation after generation of this family was, in the later stages of the disease, were strapped to a bed at night.
Records show that throughout the 18th, 19th, and 20th century, deaths consistent with FFI run down through the generations of this family. Then in the 1980s, a descendant of the family named Silvana suddenly began showing symptoms of the disease. This handsome, vibrant playboy had lived with the shadow of this potential killer all his life.
Silvano looks as if he's sleepwalking, but that is really because he's in a permanent state of pre-sleep behavior.
Silvano and other family members were filmed at the University of Bologna's sleep clinic.
Often you will see him and other patients performing gestures like combing their hair or washing their hands or handling objects.
Silvano and the others were unable to drop into a deep REM sleep. Sleep medications only accelerated their restless descent toward death.
We gave an intravenous doses of barbiturates in an attempt to help the patient sleep. The result was that they went from this pre-sleep, non-sleep, to deep coma without ever passing for a sleep stage.
Just prior to his death, Silvano made a selfless offer, bequeathing his brain to researchers, which finally opened a window into the mystery of FFI. When we come back, inside the patient's brain, and two sisters face their fate. Which one will find out if they have FFI? Stay with us.
Okay, so pretty compelling. Pretty compelling video, and Ashley, it's really hard to show you or demonstrate the types of motor dysfunctions that they have and the unique sort of characteristic features that they have. It's much better to show you on that type of video. And as they mentioned, Silvano was the first patient where they actually had a brain that they could take apart. They looked in the thalamus, and in the thalamus they saw all of these really aberrant inclusions, and the thalamus looked really spongiform. And that was one of the implications that maybe this particular form of prion disorder actually affects the thalamus. And now we know, and we have more evidence to say pretty definitively that it is a thalamic type of prion disorder. So the big question was, why is there this pathogenesis? We don't have a really good model. This is the first study that actually tries to come up with a model for FFI, a transgenic model in mice for FFI. But there are some issues with this model, and those are issues that I want you as neurobiologists and geneticists to be thinking a little bit about. So some of the pathogenesis, some of the phenotypic expression, I want to also bring to your attention. And some of this is similar to what we've looked at in the last lecture. When we're characterizing prions, we need to characterize prions in a very definitive series of ways. And one of the things that we have to do is to demonstrate that there is some protease resistance. And this proteinase K resistance that you can see here actually is really somewhat interesting. So when they put in the mutant prion that resembles the prion that you find in fatal familial insomnia, so yes, they see a large insoluble mass. We also see that it is fairly protease resistant. And then when we look at, specifically at the prion transgenic mice that we have here, and we add proteinase K to it, that we have a resistant band. So the rest of the proteins have been digested away. We don't see them. But when we go back and we stain for prions using an antibody known as 12B2, and that's something that I've highlighted there for you. So you should know that antibodies against 12B2 as well as SHA-31 are how we recognize in sections as well as on Western blots that we have a mutation that is protease resistant in our prions. And you can see that we have this with the CJD isoform as well as in the FFI isoform. Again, the size is a little bit different because you also digest with a PNGase, which deglycosylates all the proteins, which makes it run a little bit smaller. Okay, so it looks like that when you have this in a transgenic animal, that it is a protease resistant form, just like we'd expect. The biochemistry looks right. That it is also showing that it's very similar to the isoform that we see in CJD, which again, the background of these mice, when we take a look at them, we want to compare FFI to CJD because we know that they share many characteristic features. And as I've also pointed out, these are antibodies I want you to know are usually used as a diagnostic feature of looking for prions associated with things like FFI as well as CJD. Okay, so we know from the video, from the lectures and everything else, that one of the key features about FFI is a disruption in sleep patterns. So you've just created this transgenic animal that has the same mutation that we find normally occurring in FFI in humans. And we want to know, is the sleep-wake patterns in these mice compared to control mice also going to be affected? The other thing to make note of, do we see some of the other neurological signs and symptoms? I just showed you that in humans, we start to see motor effects. They can't walk really well. They have a shuffling gait. Do we see the same thing in our mice that we do in humans? And again, one of the things that I'll point out to you now before we get into the actual slides is that what we call sleep architecture, how you fall into like REM sleep from like non-REM sleep, and then how you go back to being in the awake state, we have a very defined way that we go to sleep. So we go through like non-REM into REM, back up to wakeful periods, etc. And this organization and architecture of sleep is affected in these mice. So is the motor activity. They're also going to exhibit signs of motor deficit, as we'll see in a few moments. Just keep in mind one thing: mice are nocturnal. If you're working in a lab, it's a really sort of artificial type of experience that you have with mice, because you're used to dealing with them and you see them moving around. But most of the time, when we're looking at mice and we're looking at them just in terms of their behavior, they're much more active at night than they are in the day. You're more active during the day than they are, so you do your experiments and they look like they're awake and moving around most of the time during the day, but in fact their preference is to be awake and moving around during the evening, more so than during the daytime. So when we take a look, this is what we call an EEG and then the resulting hypnogram that you see down here. If we take a look at an animal that has the same prion protein mutation on the left, so it has the same FFI that we saw earlier, the same mutation, and we see it over here on the left, and we're recording an EEG, one of the things that you'll see happening is that we don't have a sort of normal sleep architecture. So they'll be in the wide awake. So this is how you would read sort of a hypnogram. So you would see that they go through NREM sleep, they kind of wake up, they stay awake for a little while, they go back into NREM sleep, they go and wake up again, they'll stay awake, and then instead of passing through NREM, they'll just go directly into REM sleep. That's very unusual. So normally what you would expect, especially at night, is that they would be in periods of NREM sleep, then they go into REM for a while, and then they come back out into sort of a wake state once it's in the evening time. And one of the things that we know is that their architecture is dramatically affected. And these are the FFI mice over here on the left, and these are normal mice to compare with. And you can see that the overall architecture of how they sleep and everything else is affected. Yeah?
In these mice, they do exhibit REM sleep, though. I don't know if are they not supposed to have REM sleep?
Okay, so that's a good question. Should they have REM sleep? They are not supposed to have REM sleep. Okay, but just keep in mind a couple of things. No, let me ask you. Since you asked me, it's fair to ask you. Do you think age of mice is important in these studies? Like when you look at them?
The same way humans.
Yeah, and so
They get trained the same way we get trained.
so is there entrainment in these mice? Actually, they don't report on that at all. Some of the mice are taken at earlier developmental stages, and some are taken at later stages, and some coincide exactly with what we'd expect in an aged individual who's around 40 to 50, and some are not. And this happens to be a mouse that's not quite at the age range that we would normally expect, but we do see disturbances in their normal sleep architecture. Again, I won't go through the entire EEG that you see up at the top. I'm not going to go through a power analysis of the different types of sleep waves. Yep, Julian?
So wait, did they do a test in the age range that we would
Yep.
expect? And
Yes.
they, like at any point throughout the mouse development, observe the lack of REM sleep?
Yeah, so later on, I'll go through different pathological features, and there is a disturbance in their REM sleep. This is just an example that they have in their data about the disruption in the normal sleep architecture. Okay. So again, if you go and see what the overall effect of sleep is, so one of the things that you can do is try to disrupt their sleep, try to keep them awake, see what happens when you are trying to keep them awake. So we know that when we start looking at these mice, that there already is a slight disturbance in their REM, so we don't get that compensation. So that sleep rebound that I talked to you about earlier on, if you stay up for a long period of time, past the time that you normally would, you tend to sleep a little bit longer. Their REM sleep is already affected, so they didn't really see the rebound in their REM that we expected. They did see one unique thing, and I don't know, I think a couple of you have actually taken HMB 420 with me before when we talk about anesthesia. One of the things that you see when you start undergoing anesthesia is so you see this really aberrant bursting behavior of neurons. And this bursting behavior is really atypical. It's not something you normally see in real sleep. You see it in certain forms of anesthesia, and you see it in certain pathological conditions. And these mice that have the same mutation that we would expect in FFI also have these really high voltage bursts, these polyphasic complexes. So it's not a nice sort of delta wave, or it's not a nice gamma wave or anything else. It's a sort of a mixture of all of those different waves at a really high voltage. And these types of spindle activity that they see is unique to these mice when they start getting sleep deprived. They don't really explain why they see that, but it was an interesting sort of thing that they mentioned in passing. So just keep in mind that this is, at least for the time being, until someone comes along and does this experiment again or creates another type of FFI mouse, that this seems to be a characteristic feature associated with these FFI mice. Again, as they get older, these mice start to exhibit progressive neurological signs and symptoms. So they develop ataxia, they have problems moving, they have abnormal flexed posture of the hind legs. Do any of you know what kyphosis is? Anyone? Yes. Yeah, an abnormal hunching or curvature of the spine. So they have this kyphosis, and they have an abnormal foot grasp reflex as well. So they do definitely start to have the same sort of neurological symptoms that we saw in humans as they start to get older, especially once they get to about 200 days of age. Then they start to see these sort of neurological signs and symptoms, just like we do with in humans. And as the disease progresses, these mice also start to lose weight. Eventually, you have to sacrifice the mice because they can't feed themselves. They stop eating, they stop food intake. And again, we know that autonomic behaviors are affected in individuals that have FFI as well. Yeah, Danny?
Do we, when they create these transgenic mice, do they progress like the earlier part of their life, like albeit like 20 days, 30 days, do they have no signs of FFI?
Yep.
And it's, and
Yep.
okay.
Yep. I'll show you that in a minute. Hopefully, you'll believe that that's true. So how long, by the way, do mice live? So some of you work with mice. How long do they live? Your lab mice, yes? About 2 years, right? So this is just over a year or so, sort of in their middle age-ish. So they start a little bit earlier, and by the time they're over 365 days of age, or 1 year, which is sort of their middle age where they're in their full adulthood, this is when they have severe problems. They can't eat anymore, they don't feed themselves. Some of it is just because they can't get up on their hind legs to be able to eat as they normally would. So you were asking about this, Danny. So if you take a look at the age range that they looked at here. So you start off with really young mice. They have the genetic background. They have the sort of FFI point mutation in the background of the CJD. And if they're between 50 and 70 years of age, 70 days of age, sorry, and you are doing a rotarod test of motor function, they stay on the rotarod just as long as a normal mouse would, okay? So we don't really see these effects until they start hitting at about day 111. And then we start to see a slow, steady, progressive, and statistically significant decline in their motor functions. Do you guys all know what a rotor rod test is, or a rotor rod assay? Can anyone explain it really quickly in like 2 sentences? A rotor rod assay for motor function? Yeah, Danny?
You just gotta balance on something and then it's moving and then they fall off when their behavior gets worse.
But that could be like anything. So it could be like on the back of a car and like you move the car. So there's this little pole or this little no, I can't describe it either. It's a bar. It's like a bar or a log, and this bar is rotating, and the mouse has to stay on this bar as long as it possibly can. And you used to do that as a kid. Hopefully the bar was a lot bigger than the one the mice use. But when they start to fall off, it's because their motor functions aren't able to keep up. It's like being on being a lumberjack and on that log spinning in the water. At some point you fall off, and it's like them here, they're falling off the rotor rod. And you can see that there is a big difference in terms of having this FFI. If you are homozygous for it, you have severe motor functions which are statistically significant. And again, it is a real indication that it starts off, they look normal to begin with, and then when they reach at about 111 days, we start to see statistically significant changes in their overall motor function. So the other thing that you saw from that video, which is one thing to talk about and then to actually see, these individuals have severe with their cognition. So we didn't see it in these series of videos. If you want, you can look it up online. And again, if you're looking for Michael Cork, if you search for him, you'll see some of the hallucinations, some of the cognitive deficits. Obviously, you can't test for hallucinations in mice, but you can see that there are problems associated with long-term recognition, cognition in other words, and spatial working memory in these mice that have the FFI point mutation associated with them. And again, one of the things that you can do is to use that novel object recognition. Do you guys remember that from earlier on? Can you, again, in 1 sentence, tell me what novel object recognition is? What would I do to test for novel object recognition? How do I discriminate whether the mouse recognizes if there's a new object or not? What sorts of things do I do? Yep, Lauren? Yep, so how much time do they spend with the new object versus a familiar object, and are they able to discriminate something they've seen before or not? Can they recognize that? And the mice that have the point mutation that's similar to what we see in FFI, they don't have that ability to discriminate a new object from an older object. If we also look for spatial working memory using a radial arm maze, and in this case an 8-arm radial arm maze, okay? In this particular case, when they've already started to exhibit neurological signs, they started to test for this. Okay, so but that's also a really good question. Keep that in mind for the time being. This is sort of a radial arm maze. Do you guys remember how a radial arm maze with 8 different arms works? How does that work? Yeah, Danny?
There's just 8 arms. One of them has something that you want, and then it's like you kind of that's the way you have arms. So the way you fall on one of the arms has a hole in it, and then you can fall and grab what you want, and the other one don't.
Sure, you could do a radial arm maze that way. So there's usually a reward associated in some of the arms, not all of the arms, and the mouse has to remember which arms it already has checked out. And so one of the things that you can do is, do you see it going back and checking if it's already looked in that arm before? It has to remember, like, I've already gone into room 2106, I shouldn't go back there, it's a waste of my time, and I'll go to all of these other rooms instead, hoping that I'll be able to get some food. And the mouse that has this mutation doesn't do so well. It doesn't have that capacity to remember, and it makes more errors. It goes back and revisits the arms that it already has before. The other thing, and as Kay was mentioning, this is actually a really important thing when you're criticizing or evaluating some of the results. So one of the things that this radial arm maze is it's also somewhat dependent on time. You have to allow it to go, but it can't go indefinitely. And so Kay's question is, are these mice ones that are already showing deficits? They were, But when they're starting to show deficits on staying on a rotor rod, do you think that it also has an effect on their movement? Yeah. And so if this is a time-dependent sort of maze that you have to take into consideration, and you have to be checking out these 8 different arms, do you think these authors took that into account? Yes or no? Yeah, actually, in this particular case, they did a really good job of explaining how they took into account that there were motor deficits associated with these mice, and as a result, they allowed for a certain amount of extra sort of leeway in terms of their assay for this recognition task. And again, we won't worry about latency so much here, just the fact that we have 2 different tests: novel object recognition, spatial memory tested by the 8-arm radial maze, and the memory impairments exist in the FFI mice, okay? So what sorts of brain areas are affected in these FFI mice? And so I love this paper primarily because it also did MRI. So it's not just histology. They did MRI in these mice. And one of the things that they showed was that in the mice that had FFI, that the thalamus, an area that we know is affected in individuals with fatal familial insomnia, their thalamus is already starting to show signs of degeneration. It's smaller. Okay, so we know that on an MRI, it's already smaller. The cerebellum, which we know is sometimes affected in FFI, I didn't go into that in detail, is also being affected here. And this is statistically significant. It looks like there's a trend in the hippocampus to being smaller, but it is not statistically significant, nor are the changes that you see in the prefrontal cortex, although there is a slight indication that with FFI, there might be some animals that are not doing as well in terms of the volume of the prefrontal cortex. If you take animals just before you have to sacrifice them, so just around the time where they're about a year old-ish, when they're not able to feed themselves, their whole brain actually is about 12% smaller than controls. So it does suggest that there are bigger sort of volumetric changes that are going on, but the areas that they know are definitely being affected in these mice, not at 80 days of age, but toward the end, is really the thalamus as well as in the cerebellum. So are the brain areas that we think should be affected? Yes, they are. Do we see disturbances in sleep? Yes, we do. Do we see disturbances in memory? We do as well. Biochemically, we also know that they are affected in these mice as well. So just again, point out this antibody. Some of you have dropped by to chat a little bit about immunohistochemistry. So this is actually a pretty good immunohistochemical stain. And I don't know if you do this in your labs anymore. You used to do this in your 3rd-year labs looking at beta amyloid plaques. This is a normal mouse brain that does not have any aberrant forms of the prion protein. It is stained at the same time with the same antibody at the same dilution. And you can see that the overall staining, as determined by the brown stain that you see here, is actually significantly less. So in the brain, throughout the cortex, in the hippocampus, and parts of the cerebellum that you see here, and other areas in the brain, that we do see a lot of this aberrant PRP, the mutant form of the prion protein, that we detect only using this 12B2 antibody that detects aberrantly folded proteins, prion proteins in these mice. So just to highlight some of the differences that they report, and we won't make a big deal of the sorts of things that they see. One of the things that they report is that within the cerebellar cortex that you see up here, that within the cerebellar cortex, the overall prion deposition was diffuse. They didn't see it really concentrated or in clumps, in other words. And that when they took a higher power view down here in I as well as in J, they saw that the overall staining pattern, the darker brown that you see here, was actually in this strip-like pattern, and it was more dot-like within the trigeminal nucleus. So they saw all sorts of different staining, but the major take-home message from this is that overall, these prion, the mice, the transgenic mice that have the prion protein that's associated with FFI, we see dramatically increased expression of the aberrantly folded protein as determined by the 12 HMB2 antibody following proteinase K digestion. Just as we would expect on a Western blot, we see the same thing in situ with this particular antibody. So this was the really cool thing for me too, not just the MRI. This is really hard to do actually. I don't know if any of you have ever worked with EM, but doing EM sections is actually quite challenging. And to be able to get really good EM micrographs like you see here is also really challenging, and they did a fantastic job of also doing a 3D stack on an EM section to be able to come up with this three-dimensional tomogram of the really unique sort of ER Golgi structure that they were seeing only associated with the mice that had FFI with it. So a couple of really interesting features that I'm just going to point out for you. So when we take a look at mice and the brains of mice in the hippocampus and in other regions that come with the FFI prion protein, one of the things that we see with this particular mutation is that we have an increase in the abundance of these autophagosomes. So something is causing the whole degradation pathway and the whole translational pathway to be affected. And I'll get to the translation part in a second, but these autophagosomes are increased and dramatically increased, as well as the lysosomes associated with the autophagosomes. So the overall amount of protein that's being turned over is really dramatically higher in these mice. The other thing that is really unique is if this is the nucleus and this is the nucleolus, in the perinuclear region, they're also seeing on this EM these little dots that you see here, which are indicative of lipofuscin. Have any of you seen lipofuscin in any of the other papers that you might have read? Do you know what lipofuscin is? Have you ever heard of a motor neuron disease like Batten's disease? No? Any transport diseases that you're aware of? So lipofuscin is one way that we can mark lysosomes, and these lipofuscin deposits in these lysosomes that we see within the thalamus suggests that it is quite it's degenerating. In other words, it's not a healthy neuron. So if you see lots of lipofuscin, it's usually found in older neurons undergoing oxidative stress, disease neurons that are undergoing neurodegeneration. And in the thalamus that you see here in an FFI transgenic mouse, we see lots of lipofuscin, much more than we would normally see. And this is sort of the overall change that we see in compared to an age-matched control mouse. We see lots more of this lipofuscin, which is a lysosomal marker for a degenerating neuron or a neuron that is severely compromised. So again, the other really cool thing is this is what a normal Golgi stack looks like, but in these mice, the Golgi bodies look very, very unusual and has this onion-like appearance where you see sort of these rings instead of these stacks that you would normally see in the neurons of the mice. So it suggests that there are two things that are going wrong in this mouse. One, there's something wrong with the ER to Golgi. So it might be that prions are accumulating and the Golgi are responding by forming these really unique looking shapes. They're not sure why. They can only identify the phenotype. And in addition, it also looks like these mice have issues with their transport as well as degradation. So the degradation machinery like the phagosomes and the phagolysosomes all suggest, along with lipofuscin, that the amount of protein turnover in these cells and in these neurons is actually really high. And again, it's not a definitive proof, but it suggests, just like our original theory, that maybe there is something going on with the normal transport of proteins following FFI. So this is more or less a summary of what I've just gone through. The EM microscopy shows that there are some abnormalities, including autophagosomes, an increase in autophagolysosomes, increased deposition of lipofuscin, and a change in the Golgi complex. We only see this, by the way, in the mice that have the FFI mutation. We don't see this in age-matched controls at all. So it's kind of an interesting test. So the last thing I'm going to mention to you, and they did this sort of as a control, and I don't think they actually were expecting the results that they got. One of the things that we know for a prion to actually be a prion, it has to be transmissible. So if you have an aberrantly folded prion, and they've demonstrated they have a protein-resistant, biochemically defined prion in these transgenic mice, that you should be able to take these prions in these mice and then transmit it through intracerebral injection into other mice. Do you guys remember that from the last lecture? So they try to do this. Again, when they try to do this in mice that are really, really sensitive, they should be responding. So you get these mice that overexpress the mouse version of the prion protein in the second bullet point, these transgenic TGA20 mice, and these are really, really sensitive. If you intracerebrally inject a prion here, you should expect to see aberrantly folded prions after the injection. However, when you take brain homogenates from this FFI transgenic mouse that has all of these biochemical features, behavioral features, changes in the ultrastructure of the neurons, and then you inject that prion in there, nothing happens. So it's not transmissible, which was, again, something that they were quite surprised by, and they didn't develop any developmental neuronal dysfunction. I'm not going to go through this in a lot of detail. If you're interested, I can give you a link for this. It's a different way to do this biochemical test. One way to test to see if a prion is transmissible is to take a test animal and inject in a prion sample. Another way is to do this biochemically using what is known as serial protein misfolding cyclic amplification, or PMCA, and it's all done in test tubes. You do serial dilutions. Every serial dilution, you should be able to still see aberrantly folded prions, and you can seed it with an already misfolded prion or an RML seed, which is what the second bullet point is. Take an already misfolded prion protein, put it in there to make the reaction faster. When they did this for this particular mouse, no change. And so they're not really sure of the entire mechanisms, and this is what the PMCA assay actually looks like. If it works and you have a real prion protein that at every serial dilution, by the end, even if you're diluting it like 100-fold, you should still be able to detect using that 12B2 antibody, the aberrantly folded prion protein, but they weren't able to see it. Yep, Denny?
So does this, because it doesn't follow the prion rule, does it get considered a prion still, or is it a completely different, like, can there be something else that's protein protease resistant that isn't a prion that still functions the same way?
So you're asking a really good question. Should they be changing the rules for how we define a prion? They're not actually suggesting that they do. They don't have a good explanation for everything. And let me just get to the last slide. It's a great question. Should this actually be considered a prion protein or a prion disease? Because it doesn't seem to follow the same rules. So they were able to generate a mouse that has the phenotype that looks like what we see in FFI. So there are sleep disturbances, there are motor differences, cognitive differences, histopathology, biochemically, it looks a lot like what we would expect in a prion disorder. It might be useful down the road. I'm guessing that these authors are probably working on a follow-up paper for this. There is an issue with ER retention at the level of the Golgi that they were able to see, but your question is a really good one, and they don't have an answer for this, because that machinery that is supposed to allow for one prion to act as the template to cause misfolding doesn't seem to work here. And we don't know why that sort of heterogeneity exists and why it's unique to this type of mutation. But at least in terms of recapitulating the disease and the pathology, they did a really good job. In terms of the transmissibility, don't know. And whether or not this is a transmissible variant, we don't know, because it's really hard to say that this particular prion, there's so few examples of FFI. As the news article mentioned, there are only 40. We only have these model systems in place. We don't really know. So it might be a new model. It might be something outside of normal prions. We're not really sure yet. So those are great questions. I'm glad that you're asking those questions because, you know, as I mentioned, this is the first time I heard this was when I was in my 4th year and again in graduate school in my 1st year, I heard all about this. And at the time, it was really fascinating. To look at it 20 years later is actually not very different than what we understood about it. Because remember, Stanley Prusner really characterized all of this biochemically back in the 1980s. And by the time I got to it, it was already more than a decade old. And we haven't really made that much progress, even using all the molecular techniques that we have right now. So maybe some of you are thinking about this disorder and thinking about how you might approach it in a different way. And that's sort of the hope that we have for you as neurobiologists and as geneticists, that maybe you have a different way to look at it. Maybe you have a different approach that we haven't thought of before. Okay, so let me, again, switch gears just a little bit, get you ready for Thursday. Make sure you know which room you're going to. So we tried, generally speaking, to keep you with the same TA. And if you, the TA who marked your written assignment is the TA who's going to mark your presentation and is going to mark your final grant proposal as well. So communicate with your TA wherever you can, and then come and see me if there are any issues with things not being clear about the grading, or if you need more feedback. Generally, I try to encourage students to talk to TAs in a polite way and in a structured way with your TAs. But make sure that you know if you're going to WI 524 or you're going to our regular tutorial room. Our group is going to be split into 2 over the next 3 weeks. Make sure that you submit your presentation if you're presenting this week. If you're presenting next week, you don't have to submit it now. You can submit it the Thursday of next week to the Dropbox that will be created for you. Bring a copy to the class on a USB, or if you're using your own laptop, bring your laptop with you as well. Stick to the guidelines about the time to present as closely as possible. You want to give your classmates just as much time as you have in your presentation. The one thing I'm going to ask you to do, if you're doing presentations, please don't read from your notes. Don't read from your cell phone. Don't read from your tablet. Don't read from the back of your hand. Don't put, this was the best thing I ever saw. It was really cool until the person turned around. Don't put your notes on the back of someone's jacket, which I thought was really ingenious until the person turned around, and then I realized what was going on. Try not to. I know it's really tempting to do that, but you'll get better marks if you're presenting just from what you have put together as a presentation. Okay. Do you have any? Yeah, Julian, do you have a question?
So by not read from notes, do you just never refer to notes at all, or just don't?
Yeah. So I think the general rule of thumb is, we've all seen this, right? Yes. So as you can see on this slide here, on figure 1B, that that's not good. Like referring to your notes once in a while and just double checking on something, that's okay. But I still give my TA, not the ones that you're dealing with now, but a TA in another course. When he was an undergrad a couple years back, he literally brought his entire laptop up to the front and he read off his laptop, because I told him you can't read from notes. So he took it as, I can read from my laptop. You can't, right? So try to have notes, but refer to them, but don't read from them. Is that fair? Okay. And I know you guys will all do great. You guys have been doing great in your midterm, your written assignments and everything else. And I'm going to try to make sure that your presentation marks also go up in a timely manner for you as well. So be on the lookout for office times. I do want to meet with all of you, as I mentioned from the outset. So good luck if you're writing HMB 320, and you know, really get those different blood vessels and stuff under your belt.
I think so.